Alrighty guys, welcome. This is Mr. Aragon doing the meiosis shelf work. I told you guys that I was going to be helping you guys with it. Hopefully this helps just a little bit for those of you guys that are kind of stuck. The first thing that I will talk about before we even get started is that meiosis, and sorry about the resolution, I'm in coffee shop Cincinnati and this is my desktop, or my laptop. So what we will discuss beforehand is we have meiosis and then we have mitosis. So mitosis is making an exact copy of your cell. So when you cut yourself, you have to make another copy of the same cell. Meiosis is it's not completely different, but it's somewhat the same, except instead of making an exact copy, what you're doing is you're making sex cells, right? So sex cells. So meiosis, two S's, you're making sex cells, right? So you're starting with one and you're making sex cells. We hopefully went over a couple of things on Wednesday. Last time I saw you guys was that in a karyotype or in a typical cell, you have 23 different pairs of chromosomes inside of the nucleus. So this is one cell, and see this little circle inside of this larger circle? This is the nucleus, right? So I know there's 23 chromosomes. I'm not going to draw 23 because my pencil is not that fine and my drawing abilities are not that great. So bear with me here. So what we're going to do is we're actually going to talk about one chromosome. We're going to observe a chromosome, basically go through the process of meiosis, right? So obviously meiosis and mitosis are the same, except for the fact that you do prophase, metaphase, anaphase, telophase once, and then you do it again. And then what you end up with is, instead of ending up with 2 cells, you end up with 4 cells, right? So you have either 4 egg cells, these are eggs, women have eggs, or you have 4 sperm cells, right? So let's get started. And there is a PowerPoint on this online, and then on Drive, and then there's also a video online as well that's posted onto your work plan. So we're going to start with a lonely chromosome. Alrighty, let me see if I can draw it up here. Alrighty, so this is a chromosome. Remember, you have a chromatid, which is 1, 2. So a chromosome is this whole thing here. It's made up of chromatids. Keep in mind, you get this guy right here, this half, from your mom. You get this half from your dad. And then before anything even happens, during prophase, what happens is you make an exact copy of your chromosomes. So I know I said you have 23. In this case, I'm only going to use 1 to model. But you would make an exact copy of it, right? So let's see here. We're not going to copy it red. We're going to copy it blue. Let's just say it's blue, right? So blue, bam, bam, right? So now you have an exact copy of the chromosome. What happens is it undergoes metaphase now. Metaphase, everything meets in the middle. So what happens is these chromatids line up. So it begins to line up here. There you go. And then what happens next is anaphase. So they begin to be pulled apart. So as Aria quoted, Anne Frank was pulled away. So anaphase, they slowly start to be pulled apart. And the cool thing about anaphase is they could be pulled in either direction. So not all the reds have to go to one side, and all the blues have to go to a different side. So let's model that actually. So one blue could go over here, or one red could go over here, another red's going to go over here. And then you also have, let's not forget about our blue friends here. You have a blue here, blue here. All right, they get pulled away. And then what happens is telophase. Telophase basically is when they start to pinch and they produce 2 cells. So these 2 poles are going to pull. And then what's going to happen is you're going to get this nice, don't laugh, you're going to get this nice little thing called the cleavage furrow. So it starts to pinch down the middle. Hopefully you guys can see that. So it starts to pinch down the middle here. And then what you do, you end up with is 2 different cells, right? So now you have 2 cells with the chromosomes, or the chromatid still in there. So you have chromatid here, the red, and there's obviously a blue and a blue. And what happens is, once it's here, it goes through the exact same phase. So it's going to go through metaphase again. Everything meets in the middle. Anaphase, they get pulled apart. And then telophase, they start to pinch, right? You make that cleavage furrow. Or I think on your worksheet, it's called a cell plate. So it starts to create a cell plate. And then what happens is it gets pinched off. So you'll end up with this red guy, or the blue guy over here, this blue guy over here, the red guy over here, red guy over here, right? So now you have 4 different cells with half of the DNA in each of these cells. Because if you remember, we started off with 1 full chromosome. This is half a chromosome, right? So if we go back to the very front, see how we started off with that, right? 2 reds, so that's a chromosome, and we ended up with half a red, half a blue, half a red, and half a blue as well. So the reason behind this is when you have sperm and egg cells meet, they each carry half of the DNA. So let's just take, for example, you have a sperm and you have an egg, and they each carry half of a chromosome, right, or a chromatid. So what happens is when they meet, they combine and they basically create a full chromosome. There we go, right? So you create a full chromosome, and now you're ready to start dividing and making a new baby, right? So you end up with a full chromosome. All right. I'm going to go over this again during class, but this is basically all you need to do for your shelf work. The only thing that I do ask is that you give me a quick little sentence over here underneath every single step to basically tell you what's going on, right? And it could be as simple as prophase, the chromosomes make an exact copy. Metaphase, everything meets in the middle. Anaphase, the chromosomes start to get pulled apart. Telophase, you start to form this cleavage furrow or this plate. And then you do the exact same thing again, right? Metaphase again in the middle. Anaphase, they get pulled apart. And then telophase, you have that cleavage furrow that begins to form. Hopefully I can get some slides from Ms. Schaefer to have you guys look underneath microscopes and actually see a plant cell while it undergoes meiosis.